Well, we're going to be talking about the study of very small things such as microorganisms. We're going to have to talk about the equipment that we use as well. So microscopes. So many, if you all know, they have this base, and they have like an arm, and they also have this part here with the ocular lenses. And then you have a stage. This is what's called the stage. And then you also have your objective lenses of different magnifications. You also have your condenser, which for here controls how much light is being put in. It doesn't actually magnify, however, but it does control the light. So condenser, we have an arm, your objectives, and then your ocular lens, as well as your stage. So that's just a basic microscope. Okay, so a little bit of history. In the 1800s going into the 1900s, so we're just going to say like 1850s to the early 1900s, we have this physician named Hans Graham, and he was looking at a staining method for different kinds of bacteria. So what he found that they had gram-positive bacteria as well as gram-negative bacteria. This had to do, of course, with the amount of peptidoglycan in the cell wall of the species of bacteria. And on the gram-positive, when he was staining them and looking at the organisms under the microscope, they are, of course, purple because of the increased peptidoglycan layer. However, the gram-negative species remained red in his staining method because they did not have that thick peptidoglycan layer at all. Alright, so the structure of the two cell types. So in microbiome, we look at prokaryotes and eukaryotes. Again, one of these will have membrane-bound organelles, the eukaryotes will. Okay, so prokaryotes are smaller, yet they have high surface area. This allows for a lot of reactions to take place. You could take in things easier, so we're going to just draw something going into the cell. You could also take things out easier, too, as well as you could also multiply. So this image right here shows a bunch of prokaryotes just having a field day. They're all multiplying very rapidly because of their smaller size. Eukaryote cells, however, are much larger. They're more complex. You have all these compartmentalization, as well as all this taking place within organelles. A lot of cellular processes are happening. So you could see by these lines inside the eukaryotic cell. And they also have a nucleus. You can make this darker area a nucleus. Alright. Microscopes. In general, we're going to be talking about the light microscope. That will go up to 1,000 times magnification. That's the one we drew here earlier in this little art project. That one, that's a light microscope. We also have the electron microscope. It's even more powerful. This goes to 100,000 times. Then we also have the atomic force. This is a new one. I've never heard of the atomic force one, actually. And what this one does, it produces images of atoms on a surface at a very magnified realm. Alright, so let's start talking about the light microscope, and we'll start talking about the others in other videos, possibly. Okay, so light microscopes. Let's just write light here. Let there be light, you know? Okay, so light will pass through the specimen and then through the several types of lenses that we have. And bright field is going to be the most common type. We again have the light source, the ocular lens, the stage for the specimen, the iris diaphragm, the condenser, which focuses the light. It doesn't magnify again. It just focuses the light. We also have something here called a rheostat that controls the brightness, and the objective lenses are right here. Okay, let's dive deeper into the light microscopy principles. Right, so magnification. What is it? Very good question. It is the apparent increase in size, so I'm just going to write that. And again, condenser doesn't magnify. So it doesn't mag. We have two types again, objective and ocular. And when we multiply these together, this creates the total magnification. For our purposes, the ocular lenses are always 10x, while the objectives could be 4x, 10x, or 40x, all the way up to 100x, depending on the model. So this means the total magnification for a light microscope would be this 100x times 10 to get to our 1,000 times actual size. Resolution, another important principle of magnification. Think of resolution kind of the partner of magnification. So resolution is also known as resolving power. And it's the minimum distance between 2 points where you could tell that they are separate. So, for example, the maximum resolving power of a light microscope is 0.2 micrometers. So, let's say we have two bacteria cells right here. There's cell 1, cell 2. Under a light microscope, I could tell that there are two separate cells at this distance. That's the max resolving power for a light microscope. The immersion lens, so remember we were talking about that 100 times 10 to get to our max magnification of 1000x on the light microscope. So in this case, we actually have to use something called the immersion lens. This is again that 100x objective lens that's on our device. In this situation, we would use immersion oil, so I'm just going to write im oil. And this special solution has the power to displace air between the lens and the specimen. And it has the same refraction as glass. This prevents the refraction of the light from going just into the air. So instead of going, let's draw a diagram. So here's the lens, here's another lens here, and here's the specimen. I'm just going to write SPEC for specimen. And here's our light source. Okay, so with the oil, so here's our oil, O for oil, and then there's nothing on this side. It's just empty space. Okay, so since the immersion oil has the same refractive index as glass, this will prevent the light from refracting into the air and missing that objective lens. So without any oil, this is what would happen with that 100x objective lens. So light would be going through the specimen, of course, but it's just going to go outwards. It's not going to hit that lens more often than not. It's just going to go like that. It's going to miss. However, if we put that oil, since it does have the same refraction index as glass, this will allow the light to be directed more often than not into the lenses once it hits the specimen, once it goes through the oil. That's why we use the immersion oil in that step. Another principle that's very important to talk about when talking about using the light microscope is contrast. How different are the objects compared to the background? So maybe something like this, let's say the bacteria are these dots. That's little contrast, but if we had something like this, those are microorganisms, that's a ton of contrast. It's again how easily the cells can be seen. Most bacteria will lack contrast. There'll be these gelatinous kind of ghost-like shapes, kind of like that as well. So, how do we increase the contrast? We have to use stains. These will dye the organisms and certain parts of the organism so they could actually be seen against the colorless background. Some stains, such as super vital stains, will really increase the contrast. So this would be like, okay, so here's a bacterium, usually colorless. If we add a super vital stain, we do that, now we could really see it. However, it does kill the organism. So super vital stains, while they increase the contrast by that much, they really let it pop out. They also kill the organism at the same time. Let's get rid of that or move it over there. Right. So how do we observe? We need several techniques to prepare our samples to increase contrast. That's the whole part. That's the whole point of all these preparations. One way that we could do it is by a wet mount. And this is for living organisms. You just basically take a slide with a liquid sample of the organism, and then you put cover slip, and then on certain light microscopes you could actually change the contrast, and then you could then get an actual image of whatever you're looking at pretty easily. However, there are also some other types of light microscopes, such as dark field, where instead of shining the light through the specimen, so if I'm going like that, and then here's the specimen, instead of doing that, what dark field microscopy does is it directs the light at an angle. So you could see the light coming in, and then we could put one of those angle symbols there. That's what's going to be happening. It's hitting at an angle. Only the light scattered by the specimen enters the objective lens. So this is our objective. So only a light scattered by specimen will enter the lens. And so the result of this would be the cells appearing as bright objects against the very dark background. So here's the dark background, and the cells would be like this because they've displaced the light this time. Alrighty. We also have other types of light microscopes, such as phase, contrast, and these play around with the refractive index of the cellular material as well as the surroundings. So dense material will become darker. So again, it's just separating the plain background in a sense and making the specimens themselves kind of pop out with this like 3D look. That's what phase contrast basically does. We also have differential interference. And this is similar to phase contrast, but this time it separates light into two beams, and they pass through the specimen, so again, specimen, and then they recombine. What happens here is an actual 3D image. So here we just basically get a lot of contrast inside and things that are dense, they look darker. However, here we actually get a more 3D image of what we were looking at. So there's going to be shading here, as well as those darker areas being contrasted more. However, we're also going to get, kind of an increased, what might I say, like a topographical look as well, because we're looking at it through like a 3D point of view. Alright, we also have to talk about fluorescence, so let's change to a neon color to talk about fluorescence. Okay, so some materials are naturally fluorescent. Some bacteria actually do have fluorescence already. Or you could also tag specimens with fluorescent dyes, so you could also just tag them too if they aren't already fluorescent. Molecules absorb light at different wavelengths, and they emit one at a longer wavelength, and it's this principle that you could use UV and direct it onto the specimen, and then you get the fluorescence. Other things that detect fluorescence would be scanning laser, or SLM. This creates detail of the interior or intact cells. And they use fluorescent dyes here, and they bind specifically to areas of interest, to internal compounds. If I had a cell, and let's say I dye it with this tag that specifically gets the chromosome of this bacterial cell, then maybe the nucleoid here would be a different color of that dye. We also have confocal, where there's fine slices using a laser beam, and this scans across the organism. It's like a CAT scan, so basically it's a CAT scan for microorganisms. We also have multiphoton, and lower energy. So it's just basically a lower energy of this. But it also penetrates deeper. The light will penetrate deeper to get interior views of thicker structures. And it's also less damaging. Alrighty, and that's all I have on light microscopes. Please do something nice for someone and have a good one.